You're welcome to Excellent Simplified Tutorials.
Alright, you're welcome once again, and today we're going to be talking about the glycolytic pathway. The glycolytic pathway. Now, the end product of carbohydrate digestion is monosaccharide, which we can take, for example, the glucose. Glucose is a monosaccharide. Now, let's look at this diagram. Okay, let's assume this to be your intestine. Now, your mouth is somewhere here, and after eating carbohydrate food, the carbohydrate-based food passes through your gullet to your stomach. This is your stomach. From here to your small intestine, where digestion ends. Now, as digestion ends in the small intestine, the monosaccharide now is absorbed into the bloodstream. Now, what happens to the monosaccharide in your bloodstream already? What happened to the glucose? Now, the glucose can undergo breakdown, glycolysis. Now, when we say glycolysis means, you can divide this into two: glycolysis, glyco, and lysis. Lysis means breakdown. So what we are breaking down here is glucose. So glycolysis is the breakdown of glucose. Now, how do we break down glucose? Our glycolytic pathway takes place in the cytosol of the cell. And to break it down, we'll follow this pathway. First, this is what we are starting with: glucose. Glucose is first phosphorylated to glucose-6-phosphate. Glucose-6-phosphate. And the name of the enzyme catalyzing this reaction is called hexokinase. And you agree with me that here, glucose and glucose-6-phosphate, we are attaching phosphate on position 6, on carbon 6 of glucose. We're attaching phosphate on carbon 6 of glucose. So we are taking that phosphate from somewhere. Where did we get the phosphate from? From ATP. So ATP is hydrolyzed in the process, and ADP is given off. So we are taking one phosphate group from ATP and attaching it on carbon 6 of glucose to have glucose-6-phosphate. Next thing is glucose-6-phosphate to be isomerized to fructose-6-phosphate. So you agree with me that glucose and fructose are isomers, right? Now, glucose-6-phosphate is isomerized to fructose-6-phosphate, and the name of the enzyme catalyzing that reaction is called phosphoglucose isomerase, or you call it glucose-6-phosphate isomerase. Either way, you're correct. Alright, so the next thing, fructose-6-phosphate will be converted to fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate. Now, if you can see, we are attaching another phosphate. That's why we call it bisphosphate. So we are attaching a phosphate group on carbon 1 and carbon 6. So fructose 1,6-bisphosphate. On carbon 1 and carbon 6, there was already a phosphate on carbon 6. So we are attaching another phosphate on carbon 1. It means we are getting the phosphate from somewhere else. And where is that? ATP is coming in, right? Now, from fructose 1,6-bisphosphate, the name of the enzyme catalyzing this reaction from fructose-6-phosphate to fructose 1,6-bisphosphate is called phosphofructokinase. Phosphofructokinase. Now, at this step, fructose 1,6-bisphosphate is broken down into dihydroxyacetone phosphate, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Now, the name of the enzyme catalyzing this reaction is called aldolase. Aldolase. Now, dihydroxyacetone phosphate cannot continue in the reaction again, and so it is converted back. It is isomerized to another molecule of glyceraldehyde 3-phosphate. So now we have two molecules of glyceraldehyde 3-phosphate because dihydroxyacetone phosphate here is converted to another molecule of glyceraldehyde 3-phosphate. So we'll just follow one pathway. Now the glyceraldehyde 3-phosphate, okay, here we had our ATP. ATP was converted here to ADP. Now at this point now, glyceraldehyde 3-phosphate will be converted to 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate. Now there is an important point here. Glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. You can see with me that we are adding another phosphate. We are adding another phosphate on carbon 1, but this time the phosphate is not from ATP. The type of phosphorylation taking place here is called oxidative phosphorylation. Oxidative phosphorylation. So in this place, this oxidative phosphorylation is done through the electron transport chain, not from ATP. When we use ATP to add phosphate group to a substrate, we call that substrate level phosphorylation. So at step 1 here, substrate level phosphorylation. At step 3 here, substrate level phosphorylation. But here, it is oxidative phosphorylation. Oxidative phosphorylation. Now, 1,3-bisphosphoglycerate, let me take it up here. 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate here is converted to 3-phosphoglycerate. 3-phosphoglycerate. And you see with me that one phosphate group have been given up. So in 3-phosphoglycerate, one phosphate group have been given up in form of ATP. So ATP is given up here. Now 3-phosphoglycerate is converted to 2-phosphoglycerate. 2-phosphoglycerate. 2-phosphoglycerate is converted to phosphoenol pyruvate. And phosphoenol pyruvate is finally converted to pyruvate. It's finally converted to pyruvate. Good. So I want to point out something here. There are 10 steps here, but out of these 10 steps, 3 are irreversible. Now step 1, 3 and 10, this step 1, step 3 and step 10 are irreversible. So the other ones we use reversible arrows. We use reversible arrows. So step 1, 3 and step 10, they are irreversible. Now some of the enzymes I didn't mention here, glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. That reaction is catalyzed by glyceraldehyde 3-phosphate dehydrogenase. Dehydrogenase. Of course, you know it's an oxidation reaction taking place here now. Now, 1,3-bisphosphoglycerate to 3-phosphoglycerate is catalyzed by the enzyme phosphoglycerate kinase. Phosphoglycerate kinase. Now, from 3-phosphoglycerate to 2-phosphoglycerate, you can see that the phosphate group is just moving from position 3 to position 2. So the name of that enzyme is called a mutase. A mutase. Mutase is any enzyme that moves a particular group from one position to another position. So the name of this enzyme is sorry for this, 3-phosphoglycerate. Phosphoglycerate mutase. This enzyme from 3-phosphoglycerate to 2-phosphoglycerate is catalyzed by phosphoglycerate mutase. Now from 2-phosphoglycerate to phosphoenol pyruvate, and from phosphoenol pyruvate to pyruvate. Remember we said this is this last step is irreversible. This last step here is catalyzed by the enzyme pyruvate kinase. Now can you guess the name of the enzyme? From, as you can see, the enzymes are following trends. So from 2-phosphoglycerate to phosphoenol pyruvate. 2-phosphoglycerate to phosphoenol pyruvate. What do you think is the name of the enzyme? Okay, the name of the enzyme here is called enolase. Enolase. That's the name of the enzyme. Alright, if you look at this point again, phosphoenol pyruvate to pyruvate, we are not mentioning phospho again in pyruvate, meaning we've given off the phosphate group at this point. So a phosphate group is given off here in form of ATP. Good. Now, come back to the starting. We said glucose is first converted to glucosylphosphate, from glucosylphosphate to fructosylphosphate, fructosylphosphate to fructose 1,6-bisphosphate. Then fructose 1,6-bisphosphate is cleaved, is broken down to dihydroxyacetone phosphate and glyceraldehyde phosphate. Now we said dihydroxyacetone phosphate cannot continue in the pathway again, so it is converted to another molecule of glyceraldehyde phosphate. Good. Now we have two molecules of glyceraldehyde phosphate now. The glyceraldehyde phosphate is converted to 1,3-bisphosphoglycerate, from 1,3-bisphosphoglycerate to 3-phosphoglycerate to 2-phosphoglycerate to phosphoenol pyruvate, and finally to pyruvate. So this is your glycolytic pathway, and it takes place in the cytosol. The question now is, how many ATP did we get altogether from this glycolytic pathway? If you look at this, we used 1 ATP here. So we use 1 ATP here as step 1 here. Step 3, we use another ATP. So you can see that we use 2 ATP altogether. We use 2 ATP. Now let's see how many ATP we produced. Remember, the purpose why we are breaking down glucose is to get energy. But from the starting, we already used 2 ATP, 2 molecules of ATP. How many ATPs were produced? Now, in step from here, this was oxidative phosphorylation, so we didn't use any phosphate group. But here, we produce 1 phosphate group here. But remember that anything we are doing here applies 2 ways, because this is converted to another molecule of glyceraldehyde phosphate. So since it's converted to another molecule of glyceraldehyde phosphate, anything we have on one side will multiply it by 2. Now, 1,3-bisphosphoglycerate, we produce 1 ATP here. Since the glyceraldehyde phosphate are actually 2, so this is 2 ATPs. Here we produce another one, 2 ATPs also. So you can see that we've produced 4 ATPs altogether. 4 ATPs altogether. Is it not? And if we pay back the ATPs we used, we end up with just 2 ATP from the glycolytic pathway. But the question is, what happens to this pyruvate, the end product of glycolysis? What happens here? Now this pyruvate can go into the tricarboxylic acid cycle. So that you call it the tricarboxylic acid cycle, or you call it the Krebs cycle, or you call it the citric acid cycle. So we'll look at that very quickly.